Welcome, everyone, to the Complex Causes of Autism webinar series, hosted by Autism New Jersey and sponsored by the New Jersey Autism Center of Excellence. Next slide. I'm Dr. Suzanne Buchanan, Autism New Jersey's Executive Director. For those of you who don't know us, Autism New Jersey is a nonprofit that helps individuals, families, and communities. Our helpline staff connect individuals and their caregivers to reliable resources for autism diagnosis, treatment, education, and so much more. We help government leaders create autism-friendly laws, and we inform healthcare providers, law enforcement officers, and other professionals about autism best practices. Through our awareness and engagement efforts, we work to create communities where people with autism are accepted and thrive. I have here with me Cassidy Grom, our Communications Director. Cassidy will help facilitate the question and answer session during the second half of the webinar. Many of you submitted questions for our presenters when you registered, but if you have questions during the webinar, please submit them through the Q&A feature at the bottom of your screen, and we'll try to address as many as we can. And please keep an eye on your inboxes for invitations to future talks. Clearly, the community is interested, so we want you to keep the questions coming. Just a few more housekeeping notes. We will put a link to a certificate of attendance in the chat at the end of each day's session, and we will be making these sessions available on our website in the next few weeks. As always, if you have questions about autism resources, how to access services, or about our statewide initiatives, you can always call our helpline at 800-4-AUTISM. Next slide. We're here today to share what researchers know about the causes of autism and help answer some of your questions. Unfortunately, there's been a lot of misinformation about autism, so it's important that we lean on credible research and reliable resources. And we've had more than 1,400 people register to join us this week. That's an incredible testament to our community's desire to learn more about the scientific underpinnings of autism. And we know individuals with autism are very near and dear to our hearts, and so we're excited to join with you here today to learn more. Over the next 3 days, we'll explore the potential genetic causes, environmental causes, and how those impact brain structure. Our Rutgers experts will explain to you where research currently stands with their specialties and help us understand areas where we need further research. All 3 experts have extensive experience in their fields, and as our team was working with them, we learned so much. So we're really excited to dive in today and explore the latest developments in the field with you. And we're also grateful to have such a great partner like the New Jersey Autism Center of Excellence for sponsoring these talks. So now the introduction of our first presenter, Dr. Chiara Manzini, is an associate professor in the Department of Neuroscience and Cell Biology at Rutgers Robert Wood Johnson Medical School. She studied human genetics at the University of Pavia in Italy and received her PhD in neurobiology and behavior at Columbia University. She received additional training at Boston Children's Hospital and Harvard Medical School. She taught at the Institute for Neuroscience at the George Washington University. The main goal of Dr. Manzini's research is to identify genes that are essential for humans' mental processes and to find out more about the biological processes and interactions that occur within cells that may lead to muscle and nerve disorders, as well as developmental disorders like autism. Right now, her lab is focused on exploring signaling pathways within the human body and how those pathways may lead to more males with autism than females. Without any further ado, please join me in welcoming Dr. Chiara Manzini.
Thank you, Suzanne. And thank you all for coming and coming to listen to us. So just as a brief introduction, I am a neurogeneticist. What that means that I have training in neuroscience and human genetics, and I'm interested in finding what is changed in the DNA of people where the development of the brain is not typical or is disrupted. Before I start, I wanted to introduce the language that I'm going to use throughout the presentation, as it's now customary in the field. I know that there are different preferences in the community of how people are referred to, but for consistency, this is what I'm going to do, and I mean no harm. So individual with an autism spectrum disorder diagnosis will be defined as autistic person or child Male and female are individuals with XY or XX chromosomal sex, respectively. We now have a definition of very severe autism, which is profound autism, which are children that cannot speak or use very few words, so nonverbal or minimally verbal, with moderate to severe intellectual disability and may need constant 24/7 supervision. Because I know that this webinar is for everyone, I will try to reduce the amount of scientific jargon as much as I can and use like very simple words and very simple concepts. But I'm giving you the scientific jargon in parentheses in the slides, because I know that there will be people that have tried to do a lot of reading about their condition or the condition of their child. So to get us started, the diagnostic criteria, so what we use to diagnose autism, are social communication deficits and restricted interest and repetitive behaviors. But the spectrum has multiple other symptoms that are present at the same time, or different ways that the condition happens. And so you may have depression and anxiety or ADHD, but also great variability in intellectual ability and language skills. And so, as we were saying, profound autism will have severe intellectual disability and no language. And this is the type of rare genetic form of autism that we study in my lab. At the same time, I've had autistic scientists in my group. And so it's incredibly variable how autism is present in the human population. And the needs of these different groups can be very different. Another thing that we know about autism is the sex bias, the fact that for every one girl that is diagnosed with autism, there are 4 boys, which is something that the community has really been trying to figure out. So why should we look at the genes? I'm here to tell you about the genetics. So there were studies that were done in the 70s looking at identical twins, which is a simple way to see whether something should be genetic. And in those studies, they found that 60 to 90% of identical twins both have autism. When you look at fraternal twins, they have a much lower chance of both having autism, which can vary depending on the study. This told us that autism is highly genetic, but then we had to figure out in what way it is inherited by people. Before we go into this, I'm going to go to the birds and the bees and tell you how our genome is inherited and how our genes are inherited. So the DNA comes in this like long sequence of letters, and basically scientists assign the letter to the 4 different molecules that make the DNA, which constitutes what's called the sequence. So when we talk about sequencing, we're reading this very long sequence, which is in our genome, which determines whether we're a human, whether we're male or female, and all kinds of things that happen in our body. This very long string of letters is usually packed in these structures called the chromosomes. So when a baby is made, half of the genome comes in the sperm that will have 23 chromosomes from the dad, and half of the genome will be in the egg with 23 chromosomes from the mom. When the egg is fertilized, you get what is called a zygote, which has the complete set of chromosomes in 23 pairs. So identical twins, which are called monozygotic, which means from the same zygote, come from the same cell. They have the same identical genome, which is what told us that if 90% of them both have autism, it has to be genetic. Then in general, you have to figure out what kind of inheritance is happening. And so there could be one genetic change that by itself gives you up to 100% risk of developing a condition. These are called monogenic disorder. So altering one gene is sufficient, and an example of these would be sickle cell disease or Duchenne muscular dystrophy. But there are also genetic conditions where you need multiple genetic changes to happen at the same time, or polygenic, where each variant, each change adds some risk that sometimes is very small. It could be 5%, 2%. And then you need a lot of different variants that give you, in a way, predisposition to develop the disease. and increased risk. And these are type 2 diabetes, polycystic ovary syndrome, or PCOS. And this is where environmental factors can become really important to increase your risk. Then the other question is like, is there only one genetic cause? And so this one gene or combination of genes, is it the same for every single autistic individual, or are there different ones for different people? And so this is what the field was facing with when the research started now like 40 years ago. So there were some challenges that needed to be overcome because single gene and multi-gene conditions are studied in different ways. If you're trying to find only one gene, you have to look at a family. And so you need to collect the DNA from the autistic individuals and the family members and figure out how the condition tracks. If it's multi-gene, you do what is called the genome-wide association study, or GWAS, which you might have seen in the news, where you need to collect the DNA from a large number of people with a condition and a large number of controls that don't have that condition. With the additional issue is that autism presentation is variable, and we've known from the very beginning that severe autism can also be part of a more complex genetic syndrome, such as Fragile X, which has other symptoms in addition to autism. And the initial studies revealed that we needed thousands of participants for either approach that we wanted to use. And so I really like this schematic because it summarizes pretty much the past 30 years of research that have been done on autism genetics, which has been a huge amount of work. One thing was to create these very large research groups that would collect tens of thousands or hundreds of thousands of participants, which are shown here on the left, that then resulted in multiple different studies identifying autism genes. And so the first one was the gene for Fragile X. That, in a way, even if it wasn't easy, was the easiest because we knew it was on the X chromosome and you only had to focus on one chromosome. Because at the time, there wasn't the ability to sequence the entire genome, which we achieved in the early 2000s, which is when really the ability to sequence the entire genome allowed these large research groups to have bigger and bigger studies. And the initial studies really told us that the size of the number of people was not enough and we needed more. And so in the past 5 years, we've had some of these larger studies that you will see were up to 20,000, 30,000, or 40,000 autistic individuals in the study that gave us information. So to make a long story short, what all of this work taught us is that there are many different genetic causes of autism. And I've chosen this particular pie chart from the Autism Science Foundation because I think it represents what is the most interesting to you. Like, can I get a genetic diagnosis? And by a genetic diagnosis, I mean, can I go get a genetic test that will tell me what is the gene that is causing autism in my family, in my child, or in myself? And so right now we can provide an answer for around 25 to 30% of people, and 70% we still don't know. And what I'll do now, I'll explain what these different genetic changes can be. So the single gene, Fragile X, I already introduced. Fragile X is actually the most frequent single gene cause of autism. It's a form of intellectual disability with 20 to 30% of kids diagnosed with autism. It has a very specific way that the face can look, and epilepsy. And because it's on the X chromosome, it's more severe in boys who only have one X. The gene FMR1 regulates a large network of other genes. And so this is going to become important as we will be looking at all of the different autism genes that have come up from the research. So Fragile X accounts for up to 1% of autistic individuals, but performing these genetic studies, we figured out that these inherited single gene variants that you can track in the family only account for 3 to 5% of autistic individuals. And in general, they tend to be on the severe side. Another type of variant is what is called the single nucleotide variant. And so I told you about the sequence, and this is basically one letter change in the sequence that can be a different letter, or the letter can be deleted, so it's not present anymore. These are the most high confidence autism genes from a recent study. Don't try and go read and find your gene at the bottom. I will give you a resource at the end where you can look it up. But what this study did is basically look at a really large number of people. And I like this schematic because they summarized how increasing the number of people that we're looking at allows us to identify more and more genes. And so this initial study had 4,700 cases in 2015, but then as we increased the number, 20,000 cases in this 2022 study brought 72 really high confidence genes, which are the ones in the graph, but 255 other genes that we're pretty sure, but we can't say with high confidence that are associated with autism. And the way to do this, why there are more and more and more genes every time we do more people, is that we have to find changes in the same gene over and over again in different kids with different changes. And that allows us to say that this is what is causing autism. So one thing that was interesting here, I told you that the inherited variants are a very small contribution, but most of these were what is called de novo. So what is a de novo variant? De novo means a new, so it's a new change. It's only found in the autistic child on one of the chromosomes, which is called heterozygous. This variant originates in the sperm or the egg of the parent. So it is inherited from the parent, but it's not in the body of the parent. So that if you do a cheek swab or a blood draw, you will not find the variant. And the reason why these variants originate is like that as the sperms and eggs are being made, they will introduce mutations. And I think one of the questions that people had is like, is paternal or maternal the paternal and maternal genome, like responsibly involved in autism. And in this study, they actually found that the paternal age, and so the paternal age has a strong contribution to developing autism, because the novo variants are preferentially inherited from older fathers. Sperm is produced throughout life, and so is more susceptible to de novo changes because we accumulate more and more mutations as we age. And a good parallel, for example, is thinking about as you get older, you're more likely to get cancer, which is a mutation happening in your body. Another type of variant are copy number variants, where you can have a whole chunk of the chromosome that contains multiple genes either deleted or duplicated. So it's either gone or it's present twice. This is a list of the copy number variant of these changes that we know are associated with autism. Because they affect multiple genes, they're often associated with more severe diseases, and it could happen in the Angelman region, Phelan-McDermid region. One thing that I want you to take away from this is that the ones that are associated with autism, in a lot of cases, can be either a deletion or a duplication, which tells you that too little is not good, but too much of the same thing is also not good, which brings us again to the concept of balance, of the fact that genes have to be present and functional in the body at a very specific balance. And so these single gene or single genetic change causes contribute to this around 30%, where you have a fraction of the inherited, the copy number variants, and the single nucleotide variants. And so what about the rest? So the rest is thought to be highly contributed by these multi-gene causes, which are what's common variants. There are variants that are changes that are present in the population. And we're trying to figure out through these case control studies if they're enriched or if they're increased in a population with autism. And so there have been few large studies that have been able to identify some of these variants. And again, each one adds just a little bit of risk. And in fact, most of them are not necessarily inside the gene, breaking a gene or changing the function of a gene, such as the other ones that I was telling you about, but they are outside, but they regulate how that gene is active in the body. Through all of these studies, combining all of this information, so all of those large research groups that I showed you before are constantly working together and sharing the data to figure out how these variants work with each other. And what we figured out is that the autism risk variants add up. And so you may have this like low risk, multiple low risk variants that lead to a less severe form of autism. The single high risk ones tend to be more severe, but then you could have a person that has a high risk variant and multiple low risk, which ends up having more severe presentation. And this is what can explain two people having a change in the same gene having different severity. One question was always been, can the genetics explain the male bias? And here, what I can tell you is that yes and no, because we don't know yet. We know that girls need more genetic risk variants to develop autism in their genetic background than boys. And there is a lot of discussion of whether girls are protected from these from most brain conditions actually, or males are more susceptible. Because for example, even in prematurity, preemie boys will tend to have more neurological issues than preemie girls. But there was a study that just came out a few months ago looking at all of the single rare changes that have been identified, those single nucleotide variants and those de novo changes. And they seem to have the exact same risks in males and females. Part of what my lab is doing, looking at the sex bias in autism, is looking at the impact of sex hormones. And so testosterone and estrogen, and seeing how they can regulate brain function to possibly change how boys and girls are affected by autism. So this is probably a bit frustrating because you're going to say, well, great. So now there are hundreds of genes, several different types of mutation, and also all of these multi-gene causes. So why are there so many genes? Well, 1/3, so 15,000 of the 20,000 human genes are active in the brain. The brain is a very, very complex organ that has to control everything that you do. And so Dr. DiCicco-Bloom on Thursday will really explain the brain development, but I will give you the little cliff notes before you get there. And so I want you to introduce you to the primary cell that does all the heavy lifting in your brain, which is the neuron. And in this really pretty picture, each one of the balls and squiggles is a neuron in the cerebral cortex, which is the part of your brain at the top of your head with the hills and valleys. And this is just one of those cells. So each neuron has this very complex branch structure, which we call the dendritic arbor, the dendritic tree, that looks like a tree. This structure collects information. So a large neuron like this in the cortex can receive more than 10,000 contacts, each individually coming from other cells surrounding it, from other cells far away in different parts of the brain, and also from cells throughout the body that are speaking to the cortex. It has to collect the information. All of that information is integrated in the cell body of the cell, that then has to decide whether to give a response. So it's a very powerful computer. There are 80 billion neurons in the human brain and 800 trillion contacts and synapses that are active at can be active at any given time. And so to give you an example, if you're listening to this webinar, there's going to be neurons that tell you that your head is up and that your feet are down. There are neurons that are controlling your heart rate, or they're controlling your body temperature. There are neurons that had to remember where your keys were this morning when you were looking for them. And for example, if you had to learn a new language and English is not your first language, there are neurons now that are translating what I'm telling you, and that had to learn, and that had to learn a completely new language. So the neurons genes live or are produced or live in the cell body, and what they do is everything about the neuron's life. And so as a neuron is born, it has to acquire a certain identity. So it will have to know, it is the neuron controlling your heart rate, or is it the neuron remembering your keys? The location where it needs to go in the brain. Sometimes neurons are not born where they need to be, and so they do what is called migration. They have to go to a different place to be able to integrate with that particular circuit. The way that they respond to stimuli is different. One example could be neurons responding to dopamine. You might have heard about dopamine with reward and with addiction. And so those cells have to know that they can respond to that particular molecule. And then one of the most important jobs that the neuron's genes control is plasticity. So it's the ability of a neuron to respond to a stimulus and to change, which is the basis of how memory is formed. And so the genes mutated in autism have been found to classify in different groups. There are genes that control neuronal communication, synapse function, and plasticity. There are genes that function in the body of the cell that actually control the expression of hundreds of other genes that do a lot of different jobs to keep the neuron healthy and responsive. There are also genes that control how the neuron responds to nutrients and growth factors. So there can be several different ways that the function of the neuron is disrupted, leading to autism. One thing that we have found from studying all of these genes and trying to figure out common patterns is that risk genes for autism are involved in fetal development. And so now we really think that autism develops in the womb, even if it's diagnosed at 18 months, because that's what we can do with the current diagnostic methods. There is a lot of research going on trying to figure out if we can diagnose autism in babies and in much younger children. And the other thing that I want you to remember is that often these genes are only partially changed in their function. Those single nucleotide variants are only on one chromosome. The other chromosome has a copy that is working. And so it's a tweak in how the neuron work. And so I've told you there's tons of gene, the brain is super complicated. How does this help me? Like how does this affect me, my child? Well, if you are someone which has an inherited or de novo monogenic change, you can benefit from research on that specific gene. In the sense that you can figure out what is the rate of inheritance, what is the likelihood of having another child with the same mutation, because the inherited causes also affect other organs. You will have knowledge on how the condition progresses as the child gets older, and you can figure out which kind of specialists you want to talk to. And then there is the development of specialized therapies for that particular gene, with the holy grail of like developing gene therapy that can actually correct and restore gene function. And so the way that we go about it is that you find a mutation in the humans, you develop some kind of animal model or stem cell model. Usually it's going to be a mouse, and then try and develop therapeutics after you have understood what the gene is doing. Go back into the preclinical model and then go back to the human. With autism, we are doing this on a very large scale. And so right now there are labs that are comparing more than 100 mouse models of different genetic mutations, or hundreds of stem cell models with different genetic mutation, to try and figure out, can we group them in some way? Because there might be groups like the Fragile X group that is a much larger number of people, but some of these genetic causes sometimes affect like 10 people all over the world. And then what can we do for them? So can we figure out how to group them to develop targeted therapies that we can use for multiple genetic causes? In addition, because so much research is being done on the genetic models, brain changes that are found in these models can be applied to other autistic individuals. And so we're looking for what is called the biomarker, trying to find something that is changed that we can check and find in other people to be able to see if they would be amenable to a particular treatment or a similar treatment. And so to conclude, what are we still missing? What are we still doing? Well, most of the genetic research has been done in people of European ancestry. And there is still a lot of work to do on people coming from different parts of the world. Your genetic background is specific to the area of the world that you come from. And so there is a large amount of shared DNA, but also there are ancestry-specific changes. And so most of the work on autism has been done in Asia, has been done in China, where they figured out that some of the same causes are also present there, but there are also some other genetic changes that are specific to the population. And a lot more work needs to be done on Africa and Latin America. So more recruitment of study participants is needed in both the European and non-European population, in particular for these multi-gene polygenic variants, and finding these ultra rare, which are the ones that will affect only a few people. But the studies we have done so far focus on the child, and we have no idea whether the genetic makeup of the mother can also provide some risk in changing the function of the placenta or changing fetal development. And so maternal genetics is also being studied right now. These are very complicated studies because you basically need to take women when they become pregnant and then follow them for a really long time, and you have no idea whether that person is going to have a child with autism. So our study is just being done on the maternal population and then taking the people where the families where autism appears to figure out what happened during the pregnancy. And really importantly, I told you about the gene. I told you how some of them really could just present some kind of predisposition, and the gene environment interactions are extremely important. And tomorrow, Dr. Halladay will tell you all about these different environmental and physiological changes that can happen during pregnancy that strongly contribute to the development of autism. And then more comparison of the single gene causes to find patterns, which is what is being funded by the NIH right now to really try and get people to work together even more in a way that is the same in every single lab to try and figure out these patterns that we can use for treatment and for better understanding autism. And so with this, I have a few resources. I've asked Cassidy to put them in the chat. I don't know if she's going to be able to do that. The SFARI gene, you can go look up if you have a genetic diagnosis, your particular gene. ASF has this really great graphic on recurrence rates, on like what is the risk of having a child with autism if your brother has autism, if your sister has autism, etc. And also I wanted to highlight, if you're interested, the current genetic recruitment in New Jersey. The Simons Foundation for Autism Research has this very large project, the SPARCH project, that has so far recruited 380,000 people. The recruitment in New Jersey is a Children's Specialized Hospital, which is where the New Jersey Autism Center for Excellence is. And they would be very, very excited to speak with you. And so one is the link and one is their email. And they wanted me to tell you, because this is a privately funded research study, your privacy is really, really important to them. And with this, I thank you for your attention, and I hope this was helpful, and I'm happy to take any questions.
Thank you, Doctor. That was so insightful, and I'm sure everyone here learned a lot. We got so many questions from participants, as well as from people who submitted their questions when they registered a few weeks ago. So we will not have time to go through all of them, but there were many that had been submitted multiple times. So we're going to try to hit the ones that people were asking the most. So the first question we have here is, can my genetic information be used to harm me as an autistic person or my autistic child?
No, because there is a law for that, which is what I always, whenever I enroll participants in my studies, I always have to tell them. So in the US, there is a law that is called GINA, which is the Genetic Information Non-Discrimination Act, that basically you cannot be discriminated based on your genetic findings for healthcare, you know, not being able to get insurance or any other thing. Also, your genetic information is private, so it shouldn't be shared with anybody from your healthcare provider without your consent. If you're in Europe, just in case, you know, it's even more restrictive what type of information is being shared. So there are very strong laws to make sure that doesn't happen.
Thank you. Yeah, that's very reassuring information, I'm sure. We did have a question about the paternal versus maternal genes. It says, do the paternal or the maternal genes play a larger role in the inheritance of autism, or do we not know?
So it's a complicated. Everything with the genetics of autism is complicated. So paternal age, yes. So we know pretty clearly now that the age of the father increases the likelihood of having these de novo variants. The age of the mother also a little bit, but not quite as much. While when you have chromosomal rearrangements or chromosomal changes like Down syndrome, that's where they always tell you the age of the mother is what matters. But for autism, the age of the father matters. There is also some research on whether you have, because girls with autism need to have more genetic variants and a stronger genetic background of autism, it is also possible that the sister of an autistic individual may not be autistic, but may have this similar genetic risk. And so then she could potentially pass that to her child. But in general, I would say it's very rare and it's case specific.
Thank you. That actually leads right into our next question. And we had probably at least a dozen of these sorts of questions. And it's from people who have a sibling with profound autism or with, you know, they're on the autism spectrum. And they ask, are there genetic tests that they can take before getting pregnant? And then as a follow-up question, we had quite a few people ask, how do you get a genetic test to see some of these markers you discussed?
Right. So there are two different ways. So, I mean, in general, if a sibling has profound autism, they should have gotten a genetic test, or they can get a genetics test if that has not been prescribed. And so in general, what happens, you go to the pediatrician and they will call what's called medical genetics, which is the hospital department that deals with the genetic testing, and they can order the test. You can really better know what your likelihood is if you know what the variant is, what the change is in your family. If it's multigenic, if it's a combination of different genes, it's going to be a little bit more difficult. But there is some really good data in that link that I sent from the ASF that goes through all the different permutations, which I think is going to be easier. Maybe Cassidy, if you can put it in the chat, because I really love their visualizations, because it basically tells you if you have a brother, if you have a sister, like if you have a boy, because then it's also the issue, if you have a boy versus a girl, the risk is different. The risk is different, all because of these common multigenic variants. And then if you yourself want to get a test, I think you can just talk to your doctor or the doctor of your sibling, because in a lot of cases, the testing for the family members can also be covered. The issue is whether insurance will cover the genetic test. But if you want to get pregnant, you can also talk to your gynecologist before and talk about your family history and saying that you want to get a genetic test. That was going to be more successful if you know what the variant is, because those types of tests are done routinely for before pregnancy.
Thanks. And just to clarify, Dr. Manzini, it's a normal blood draw, correct? Like it's just how you would get tested for.
Yeah, you can do a blood draw or a cheek swab. I mean, sometimes nowadays they just send you a cheek swab, or they'll do a blood draw. So to just get the DNA. To do it at home, sometimes we just do saliva, but it depends on what that particular test is been designed for, to make sure that you get the clinical test. For research, if you're interested in just try to figure it out, you can join one of these large research projects. The problem with those is that it could be months or years before you get an answer. So I wouldn't necessarily use that for family planning.
Thank you. Yeah, that makes sense. There is a question about gene replacement. So it seems like there might have been some research in that in the past. And the individual who wrote in just wanted to know if there's been further research or kind of where that stands.
There has been a lot of research on gene therapy. So gene replacement would basically be gene therapy, and the way that it works now is that you take a virus that can enter the cells in your body and put the gene that is not broken back in. It has been very successful for some very severe disorders like spinal muscular atrophy, and for multiple like eye disorders. There's a lot of research in developing vectors and in developing gene therapy for some of the most common frequent causes of autism. So there is a lot of research on Fragile X that is being done right now, and there is research on Rett syndrome that is being done right now. I think at first it's going to be those more frequent causes, because we're still trying to figure out with the gene therapy. The difficulty is getting into the brain and to get enough of the brain to have the correct gene. And also, as I told you before, too little is not good, too much is not good. And so to be able to control exactly how much of the gene you give is incredibly important. And so there is an enormous amount of work going on trying to figure out how to do that. With the idea that then you could use the same approach for all kinds of different genes.
Gotcha. That'll be very interesting to see how it develops. Next question is it true that children of engineers or technicians are more likely to have autism?
I mean, there's been a lot of discussion, right, on whether highly intelligent or educated people are more likely to have autism, to have children with autism, and I kind of like stand in the middle. Even in the original study that described autism in the 40s, one of the conclusions was that because the mothers whole had gone to college, it was because the mothers are highly educated. And my response to that is like, the mother is watching that child like a hawk, and is like, and not working 2 jobs maybe, and trying to really figure out what's different. But also, there has been a lot, also these large GWAS studies that I was telling you about, they don't also exist for autism. They exist pretty much for everything, from for like like neuroticism. There's like all of these different features of psychiatric behavior that are being studied with these multi-gene causes. And so one of the thoughts is that the genetic background of the parent may already be predisposed for autism, and then the child has autism. And whether the parent is out of the spectrum or on the spectrum in a different way, it's like really difficult to say. And, you know, whenever I talk to my psychiatrist friends, it's like a psychiatric disorder. Something is called a psychiatric disorder when it impairs your ability to function normally, but we all exist on a spectrum of behavior, of people that are more social or less social, or more, you know, more detail oriented, less detail oriented, and there is genetics for that.
Great, thank you for that. Quick pause on the questions real quick for a small announcement. We know some people here today can get credit for their attendance for various programs. So for those individuals, there is a PDF in the chat link. Just download that and to submit it to the correct entity. And we still have about 15 minutes left of the webinar, but we wanted to remind you that if you guys have questions about your autism diagnosis or perhaps a loved one's autism diagnosis, you can call 800-4-AUTISM. That's our helpline here at Autism New Jersey, or you can email information@autismnj.org. So with that, we'll go right back into the questions here. One individual wanted to know, why do children with autism have higher rates of gastrointestinal issues and autoimmune disorders? Or I guess the follow-up is, do they have higher rates of these
I
issues?
mean, it has been reported, but again, it's variable, right? Because the question is like, again, what is the specific genetic cause? Because there could be some genetic conditions that also affect other organs. Also, there are neurons in your gut. And so to what extent it's the neurons in the gut that are being disrupted versus something that is happening to the gut, there's a lot of research trying to figure that out, because GI symptoms are a big problem and a big impact on the quality of life. Not everybody will have a GI issue, but also GI issues can be caused by food restriction. And so if a child only eats potato chips and burgers, that will change the way their GI system works. So it's very specific to the individual, whether it is a issue with their gut or it's caused by the behavior. And then for the autoimmune, it's interesting because it's been reported that there is inflammation, that there is systemic inflammation in a certain number of individuals with autism. But what causes it, and if any of the genes that are disrupted are responsible for it, we don't know. One thing for sure that I told you is that a lot of the autism genes regulate a lot of other genes, not just genes that function in the neurons. And so it is possible that in a different cell, they could be doing something else to increase inflammation.
Thank you for that. So, doctor, we're talking about 3 different causes of autism this week. We have genetics, environment or environmental factors, and then sort of the brain structure. In your opinion, or in your scientific expertise, of these 3, do any of them outweigh the other as a potential cause of autism?
Well, I think there's 2. So it would be the genetic and the environment that impacts brain development, right? Because those are the 2 things that impact the way that the brain gets put together. I think genetics is the starting point, and then environment is also incredibly important. And so I don't know if there will be people that have purely environmental caused autism without any type of genetic predisposition, but it's possible, which is why these gene environment interaction studies are really important to figure out what is the relative effect of the two causes. But right now we know autism is highly genetic, and the environment can have a strong impact on how it manifests.
Awesome. And that gives us a nice plug for tomorrow's webinar, which will be going over those environmental factors. So we hope everyone who's able to join us today are able to join us tomorrow and Thursday as well, because there is so much to cover. And our presenters have done such a great job of explaining where research currently stands. So back to some genetic questions. We had an individual who took a 23andMe test, and they were asking if these sort of widely available consumer tests can indicate if there's any chance of autism in their family or if they kind of carry those genes. Is that something you're aware of, Doctor?
I am not, and I would not use any consumer. I mean, I've avoided 23andMe my entire career, and right now they're selling all of the genetic information to a different company because they're going bankrupt. So for this type of study, I would really just go through. I would go through a doctor. And there are gene panels, and they can do what's called exome sequencing, so sequence all the genes in the genome to figure out if there are any of these like high risk changes. The other thing is to get it done for free, is to join one. I keep plugging the studies, but to get it done for free, to join one of these studies. And what they do periodically as they analyze your data, you can choose to have them send you back all of the information, and they will send you all of the information. So I would go through people that know what they're doing, because also 23andMe was not allowed to report on a lot of clinical features, because you need to have somebody that can explain the results to you when you get these results.
Great. And then we did have a question if there's any connection, I guess, genetic connection to MTHFR. And forgive me if there's a better way of saying that, but have you heard of that acronym before?
Yeah. So it's a metabolic syndrome. And so I think it's one of these like super rare causes where a certain subset of the kids may have autism, but it's a more general syndrome that affects like different parts of the body.
Thank you for that. And there's been a lot of talk lately in the national news, as well as within the autism community, about autism research. In your experience, do you think we are close to finding a singular cause of autism, or how do you perceive current research looking? Like, is it going to take a year? Is it going to take 10 years? Is it going to take,
I mean, one thing that we are 100% sure right now, after billions of dollars and tens of thousands of people sequenced, is that there is not a singular cause of autism. That some people may be able to figure out through genetics which gene is disrupted in their family. And then that some other people may have these like more general predisposition genes. And in a future world, right, where every newborn gets a genome sequencing, which is, it could be not that far. It says that newborn sequencing is being piloted in a lot of universities, but then the issue is like, do you want, but in a world where everybody gets a genome sequencing at birth, and can get all kinds of predictions on all kinds of disease predisposition, right? This is where some of this research is going to be interesting to like be able to identify people with this common variants. But there's also the environmental part that we shouldn't forget, that is going to be really important in different types of for different causes of autism. So unfortunately, there isn't one cause. And now what the research is really trying to do is like, okay, there's all of these different possibilities. Can we group them in some way? So that can we have a set of people that we can, if they need treatment, like treat in the same way, or if they, it's just like, to basically try and find solutions so that the very rare cases are not left by themselves.
Great answer, yeah. And we heard that echoed by some of the other presenters who will be speaking tomorrow and Thursday, that the causes are likely very complex, and there's likely multiple causes that cause different presentations of autism spectrum disorder, and for it to show up in different ways. And kind of part of that question, we had someone write in and ask if there's identical twins who both have autism, but they're on opposite ends of the spectrum, how is that possible, right? Because we know, or at least we're taught in school, that identical means identical. So is something going on with the genes, or,
So identical twins are not actually genetically identical.
That is news to me.
Yeah, no, I know. I mean, there's very little differences, but even identical twins are not genetically identical, because there are little changes. So those copy number variants that I was telling you about, these little chunks, there are some parts of the genome that can take out and duplicate little chunks. And so they're actually not 100% identical. But also their experience in the womb is going to be different, because they're two different people. And there might be one that may be slightly bigger, and the other ones, I mean, even if their genetic makeup is the same, from the second that that cell is made, the way that it develops into a person is individual. And so this is what we know, that yes, they both have autism, but depending on what happened, the amount of oxygen that they receive, any type of thing. I mean, I think actually Dr. Halladay tomorrow would be, her talk tomorrow is going to be really the one to get people thinking about the maternal environment.
Got it. Yeah, what you're talking about seems to be like the basis for a lot of twin studies, right? Where people who have maybe not exact, but very similar genetics perhaps present with autism in different ways, or present with different developmental disabilities. So yes, please stay tuned. We keep plugging tomorrow's webinar, but I'm sure it will be insightful for a lot of these questions. And then we did have another question that perhaps could be better answered tomorrow, but you might have some insight as well, doctor. The question is, having multiple miscarriages, does this raise the chance of having a child with autism? So I guess the question is, if you've had multiple miscarriages before, and then you do have a child, does that increase the chances?
I don't think so. I mean, but that maybe, Manny then on Thursday, the physician will have it. I personally don't think so. But the issue is whether there's some type of chromosomal, something that's happening with the way the eggs are being released, but that shouldn't, I don't think that should. But one of my colleagues, for example, is looking at C-section as a possible contributor to, like, an environmental contributor to developing autism, because right now in the US, everybody wants to deliver when they want, and so the C-sections are just done as a choice. And so there's a tiny little increased risk, I think, and that's what she's been working on. But multiple miscarriages, I don't know.
That's interesting. And I'm sure for patients and doctors alike, that's it's a lot to balance, right? Because some people need C-sections for various reasons.
Right, exactly. So I mean, and then what the, you know, unfortunately, it's always a question of risk, right? I mean, it's just like, is it a 10% risk, a 20% risk? It's like with everything. I mean,
Mhm. And then we are getting ready to wrap up here. So we're gonna ask a few more questions if you're willing to stick around,
Sure.
Doctor. But for those of you who need to jump off, we do just want to remind you one more time that Autism New Jersey is available to help you with your autism diagnosis or individuals in your life. We help people from birth all the way through adulthood. So feel free to call our helpline at 800-4-AUTISM to talk about a range of needs such as educational needs, clinical needs. We're here with you every step of the way. So we'll do 2 more questions and we'll wrap up for today. We had someone write in asking, are there different genetic markers for the different autism spectrum diagnosis levels? We know there's 1, 2, and 3, ranging
Right.
from milder needs to more profound. So they're curious if perhaps
Yeah.
genetics kind of line up with that.
Yeah, so in general, the single gene, those initial single gene causes, like the inherited, like Fragile X, and the single nucleotide variants in some of the genes tend to have more severe presentation. But not always, because again, it may depend on like what additional genetic background you have. And this is really the where the research is going now, that we have so much information and we can start integrating all of this information to assess individual risk. But in general, yeah, the more neurodivergent, like needing less support group tends to be more these multigenic predisposition factors.
Thank you. And we'll end on a note that's perhaps not as scientific, but is on top of mind for a lot of us. Doctor, do you have any concerns about the national funding models and the grants and the research funding perhaps being limited or taken away? What's it like on the ground right now?
On the ground, it's bad. I mean, I think the community, and it's very important, like the community really needs to come together and speak to the representatives, because that's what we do to keep the need for maintaining autism research front and center. I mean, in a way, there was a lot of excitement about this focus, this new proposal from the Department of Health and Human Services to develop these research studies on autism, which was exciting. But then at the same time, it has to be grounded in the science. And so we're all trying to figure out how to fit in that. Definitely NIH budget should not be cut by 40%. We're already on a shoestrap. Only the top 10 to 8% of grant applications are funded right now. And it's not just this administration, it was before, it's just like, we're in this situation where a lot of research that is very good is not getting done or cannot get done. And so the NIH budget always needs to increase. And as much advocacy can be done for autism to make it increase specifically for autism, as it's been done for cancer and Alzheimer's and some other conditions, is like really important. Right now, if they really cut NIH funding by 40%, it's going to be catastrophic, because then the top 5%. So you have to send 100, you have to send 50 grants hoping that you will get one. And this was really going to hurt this more rare more less studied causes of profound autism, which I think is a community that already feels they're not being studied. And I promise them that they are being studied, but you know, there needs to be more money.
Of course, and that's something our organization has spoken up about as well. You know, of course, we want more research into autism treatments and causes, but we want it to be grounded in reality and in you know, kind of common research principles, which, if you're willing, could you share a short primer on how research is done well and what it looks like to be able to duplicate research, kind of how that process looks like?
Well, you basically have to set it up, especially in genetics, in a way that, as we say, is very well controlled, right? And so, for an example, for the genome-wide association study, you need the cases and you need the controls, but your controls have to be matched in age, the same genetic ancestry, the same sex as the people that you're testing. Because let's say all of your autistic cases are tall and all of the other people are short, you're going to find genetic association to being tall, not genetic association to to autism. And so there is a lot of thought that goes into putting these studies together. And now that we're finding more and more genes, there need to be larger and larger populations. And so there's been a lot of thought in harmonizing the diagnosis across different countries, because, you know, so the scientific community is being very, very, very careful about how we do this. And so for the maternal predisposition in particular, doing a retrospective chart analysis is not going to help, because you need to collect the samples from the second the woman gets pregnant, and you need to be able to get the placenta at birth. And then maybe, you know, the child will develop autism, and then you go back and analyze. So the structure of the study has to be set up correctly for the specific question. And then, I mean, just as a point, we do not want to spend a ton of money to repeat things that we've already done, such as like the vaccines cause autism. Like, we know that there have been very, very large studies all over the world that have shown that vaccines do not cause autism, that thimerosal, you know, on multiple different components that can be in the vaccine that could trigger a presentation. And so the idea that now we want to do that again is just like, well, the money should be the money should be going to different places. And so what we hope, so I'm in one of the review panels that deals with a lot of autism grants, and on grants that on psychiatric disorders. And so as long as the integrity of the review panels remains the same and the scientists can make those choices, and can basically look at what would be the most useful, I think that would be one of the most important things. And I'll leave you with that. But advocacy is really important.
Great. Thank you, doctor. And we are rooting for you and the other Rutgers scientists, and of course the scientists who are studying autism across the world. So that is going to conclude our webinar today. I want to remind everyone to come back tomorrow to learn about environmental factors into the causes of autism. Registration is still open, so if you benefited from this today, you thought it was interesting or helpful, tell your friends. They can still register and join us tomorrow and Thursday, and we're looking forward to seeing you all again. So thank you all once again, and we will see you tomorrow.